In this video, you will get a complete picture of the metformin's mechanism of action and the effect of metformin in different body tissues such as the liver, muscle, and adipocytes. And you will see here how the metformin controls type 2 diabetes mellitus. So, metformin is a drug which comes under biguanides, and it is basically hydrophilic in nature and cationic. And so, it requires a transporter protein to get into the cell, which is OCT. In the liver, it uses OCT1 protein. And one of the noticeable peculiarity of the metformin is that it does not cause or it does not increase insulin secretion. So it is a safe drug as it does not cause hypoglycemia. And we will see the mechanism of action of metformin. Metformin acts by inducing a mild energy stress inside the cell. It mainly does this by inhibiting the mitochondrial complex 1 of the ETC inside the mitochondria. What metformin does is it causes a mild energy distress by uh, by imbalancing the ratio of the AMP or ADP is to ATP ratio by inhibiting the enzyme mitochondrial complex 1 of the electron transport chain inside the mitochondria. And this inhibition will leads to an activation of the another enzyme called AMPK enzyme or AMP kinase, AMP activated protein kinase. And this enzyme is a serine threonine enzyme which act as an energy sensor, and it quickly catches this energy imbalance within the cell by the inhibition of the mitochondrial complex 1 by metformin. And in the liver, this AMPK activation phosphorylates and downregulates the expression of two gluconeogenic enzymes, which are phosphoenol pyruvate carboxy kinase and glucose 6-phosphatase enzyme. And the downregulation of this enzyme will lead to a reduced hepatic glucose production. As well as this AMPK also inhibit another enzyme which is acetyl-CoA carboxylase, and this enzyme is involved in the fatty acid synthesis. So this enzyme, the inhibition of the acetyl-CoA carboxylase enzyme will lead to further decreases the malonyl CoA levels, and which enhances the fatty acid oxidation in the liver. So this is the effect of the metformin inside the liver. Now we will see the effect of metformin in skeletal muscle. So we all know that in type 2 DM there is insulin resistance. That means the insulin is present but the cells can't respond effectively to those signals. As a result, glucose can't get inside of the cell. That means the glucose uptake is reduced, and as the GLUT4 transporter, which normally carries glucose into the cell, does not effectively move to the cell surface. This is happening in type 2 DM or insulin resistance. So what the metformin does in skeletal muscle is the AMPK activation in muscle acts like a metabolic activator. That means like simulating how the glucose is utilized during exercise, but without exercise. That means it can mimic exercise. So in the muscle tissue, the metformin improves the translocation of the GLUT4 transporter to the plasma membrane. So what happens when GLUT4 moves to the cell membrane? It can take the glucose. So that means it actually improves the insulin sensitivity. So similar to the liver, these AMPK in muscle also inhibit that enzyme ACC, acetyl-CoA carboxylase, that which leads to an increased fatty acid oxidation and improved insulin sensitivity. Now in the adipose tissue, the activation of this AMPK downregulates a key transcription factor that involved in the fatty acid synthesis, which is SREBP1C, sterol regulatory element binding protein 1C. So similar to its action in the liver and muscle, AMPK activation in adipose tissue, in effect it promotes lipolysis, it lowers the triglyceride synthesis, and it increases the beta oxidation by inhibiting this enzyme. So now we will see the effect of metformin in the intestine. Metformin actually decreases the intestinal glucose absorption, and it increases the secretion of the incretin hormones, GLP, glucagon-like peptide hormone secretion. So the net effect is a better postprandial blood glucose control. So metformin has another effect of controlling the postprandial blood glucose level by its action on the intestine. In the above, we have discussed about the AMPK dependent mechanism of action of the metformin. Now we will see the AMPK independent mechanism of action by mainly because metformin still works effectively even in individuals where AMPK is absent. So in individuals where this enzyme is absent, metformin still shows its effect of lowering the blood glucose level, mainly by inhibiting the mitochondrial GPD enzyme, which is a part of the glycerophosphate shuttle. So by inhibiting this particular enzyme, the Metformin alters the cytosolic redox state, that means increasing the NAD plus ratio, which leads to a reduced conversion of the lactate or glycerol into glucose, and thereby suppressing the hepatic glucose synthesis through AMPK independent pathway, as well as the increased AMP or adenosine monophosphate. Increased intracellular AMP can inhibit the adenylate cyclase enzyme, which will in turn reduces cyclic AMP levels, leading to a decreased protein kinase A activity, and which ultimately reduces the expression of the gluconeogenic genes. So, we have heard about the weight loss effect of metformin. The actual reason behind this is, in the hypothalamus, the action of AMPK is just opposite to that seen in peripheral tissues. Here, the metformin inhibits the AMPK activity. In all the above cases, we have seen the activation of the AMPK lowers the blood glucose level by downregulating or inhibiting several enzymes. Here, in the hypothalamus, the metformin inhibits the AMPK activity, which leads to a reduction in the appetite and food intake, and this contributes to a mild weight lowering effect. So, in this video, we found the mechanism of action of metformin, and we have seen that the metformin comes under biguanide, and its primary action is inhibiting the mitochondrial complex 1 by causing, and this causes a disruption in the AMP ATP ratio, and the main mediator is AMPK activation, also acts through AMPK independent pathways. In the liver, it lowers the glucose synthesis and lipid synthesis, and it increases the fatty acid oxidation. In the muscle, it increases the GLUT4 translocation, it increases the glucose uptake and increases fatty acid oxidation. In the brain, or particularly in the hypothalamus, it decreases the AMPK or inhibit AMPK, which lowers the food intake and it reduces the appetite. The AMPK independent effect of metformin is through inhibiting the mitochondrial GPD enzyme as well as adenylate cyclase enzyme, which lowers the cyclic AMP levels and reduces the gluconeogenic gene expression. So, the overall effect of the metformin is it lowers the blood glucose level by both AMPK dependent and independent pathways and without causing hypoglycemia. That is the main peculiarity of metformin. It does not increase insulin secretion, but it actually increases the insulin sensitivity, and it also has a mild weight lowering effect too. So, this is the overall picture of metformin's mechanism of action. We have seen two pathways, AMPK dependent and AMPK independent pathways. And if you like this video, please subscribe to the channel and hit the bell icon to get notified for the next video.